Hello, I'm Adrian, a third-year PhD in the Duer group. Here in this group we are studying the extracellular matrix and how changes in the extracellular matrix affect the behavior of cells in normal conditions and also in various pathologies. So over time we studied pathologies such as diabetes and aging, calcification of arteries. At the moment, this group found a new interest in studying cancer biology and how cancer cells are using the matrix as a tool in order to invade other tissues. I've actually been in this group for about 4 years, and my research interest changed from studying diabetes from my master's project to studying vascular calcification as a PhD. The group has a multidisciplinary and interdisciplinary interest. Some of us are biologists, some are physical chemists, biophysicists, and actually a lot of people didn't really have much experience in this area of research before starting. Okay, so at the moment we are in our lab, and this is a containment level 2 facility designed for working with cell cultures, and I'll show you what facilities we have to offer in this specific lab. Most people in the group are doing some kind of cell work, and this is the incubator where we are growing our cells. We are growing both animal cells and human cells depending on what kind of pathologies we are studying. This is our safety cabinet where we are doing all of our cell culture work, and we actually have very clear protocols in order to prevent contamination in our cell cultures. And here on the right we have our own optical microscope that we are using to inspect our cells and make sure that everything is alright for the next steps of our experiment. We actually have access to a lot of other facilities, both in the department, in the MPAC, to fluorescence microscopes, or at the Cambridge Advanced Imaging Center in the anatomy department, where we have access to other fluorescence microscopes, confocal microscopes, and also electron microscope. So in our lab, we also have the capacity to do all sorts of assays using this instrument. So it is also suitable for people who want to focus their work on more on the biochemistry or biophysics side of things. And for our biological work, we also have 2 different kinds of centrifuges. The one at the back is a bigger one that you can use for 50 or 15 milliliter tubes. And we have a smaller one here that can be used for Eppendorf tubes. So in case you were wondering if we actually do any kind of chemistry in here, that's why we have so many chemicals behind me, and this is the fume hood where people can do some kind of synthetic work. My supervisor is actually looking at the moment to have access to other fume hoods in the department for people to do the chemistry if they are interested for their projects to focus more on that. So now I'm going to take you to where the magic happens in this group, the solid-state NMR facilities, which is the characterization techniques that we are best known for. So here is where the magic starts. On this table, here is where we are packing our samples of matrix or just cells into this kind of small rotors that we are taking them to the NMR machine in order to analyze them. This is Kinchi, one of our solid-state NMR machines. It is a 400 MHz magnet where you can use probes of 2.5 or 4 millimeters. This is the machine that is used only by our group, but we actually have access to another 400 MHz NMR machine that is used by another group as well, and we also have access to a stronger magnet of 700 MHz. Moreover, the department has great social state NMR facilities, and the people who are working for the NMR service are of great help always. Thank you for joining me in the lab tour today. I hope you enjoyed finding out more about our facilities. If you do have any questions, please don't hesitate to get into contact with us. If you'd like to find out more details about the facilities or our research in general. We are a very friendly group, so we will be more than happy to meet you.